Let me introduce the speaker of the day, welcome Dr. Prakash Sharma. He is a scientist in the ICAR Directorate of Cold Water Fisheries Research in Bhimtal, Uttarakhand. He did his BFSc from Central Agriculture University, Tripura, and he did his MFSc, Master of Fisheries Science, from Central Institute of Fisheries Education in Mumbai. And he was my batchmate as well. And he did a PhD in Texas Tech University, Texas, USA. So his specialization is in fish nutrition biochemistry, and further he did a specialization in the comparative endocrinology. Further, his PhD, he joined as an assistant director in the Directorate of Fisheries, Government of Sikkim, in 2013, and in 2014 he was in the KVK Sikkim, and then from 2015 onwards he is serving as a scientist in the ICAR Cold Water Fisheries Research Institute. So he have 23 research papers and he have 5 books published, and he is well known in the Indian circle for his skills in fish histology. And that's exactly what he's going to talk today. He's going to give an overview about how you can use the techniques in fish histology to generate data in many aspects of the fish biology, starting from general to very specific physiology and pathology sectors. So let's hear more about that story from Prakash himself. So welcome, Prakash, again. So the floor is yours now. Thank you.
Okay, Deepak, thank you so much. Can you see my screen is shared or not?
Yes, I can see your screen. Yeah,
my screen is there, right?
Yeah,
so I can start now.
Yeah, just keep the distance from the microphone. So yeah.
Okay. So thank you so much, Deepak. We'll be starting. So I'll be talking on the use of histological tools to study fish biology approach. Why I'm calling it spatial? Because can you hear me, right?
Yes.
Yeah, why I'm calling it spatial? Because this has a multiple uses and it has a spatial uses, starting from, like, if I want to study the developmental biology of the fish in terms of the gastrointestinal development, I can use this histology tool. If I want to study the ontogeny of the endocrinal system, if I want to study the environmental toxicology or endocrine disruption or microbial histopathology or anything, I can use this tool. So it has a what is called a wide spectrum application. So we can use, but this is a very old technique and very conventional technique, but it still holds an importance in the biological science and biomedical science for diagnostics and in the research as well. So why this is so interesting and why we have to use this technique and learn about this, and then how we can use this technique to study the fish biology or the fish physiology or any other aspects of fisheries research and aquaculture sciences. We'll be talking about it today. So I'd like to start with I'll start with the nutritional physiology. How did I find this subject interesting? I started this work in ICAR-DCFR in my professional attachment training during my first joining in 2015. So when I first came here, I found some of the instruments, basic instrument. I integrated those and started my work on the what is called the histology, and then a little bit of interest on nutritional histophysiology. I brought it back from my PhD dissertation. In my dissertation work, what I did was I looked into the developmental response of digestive tract in response to the thyroid inhibition or thyroid supplementation. And interestingly, what I found was thyroid inhibition retarded the development of the intestinal fold, as well as the goblet cells in the gastrointestinal tract, and supplementation restored the developmental events. And then that was my first exposure to the use of histology for studying the nutritional physiology. When I came here in DCFR in 2015, I got an opportunity to work on the developmental ontogeny of the digestive tract of golden mouse ear, and I chose to use the histology tools if I can work on it and develop the interest on how to use the tool. So while working on this aspect, the ontogeny of the digestive tract of golden mouse ear, I learned so many biomarkers of the development. So histology-based biomarkers, and those were like protein absorption biomarkers in the intestine, lipid absorption biomarkers in the intestine, and nutritional deposition and nutritional status biomarkers in the liver, zymogen granule formation, and then developmental pattern and dynamics of the zymogen granules in the pancreas. Those were very interesting things to look into. And then in that work, I could enlist around 8 developmental endpoints as histological markers. So those were only 8 histological biomarkers that I could enlist during that my first study on ontogeny. And subsequently, I got an opportunity to work with another scientist, Dr. Rakhi Kumari from SIFA. So I got an opportunity in that work on Magur, Indian walking catfish. So this species had a, so earlier species didn't have the, it doesn't have the, what is called the stomach, that is a gastric fish, and this Magur was a gastric fish eventually. And then in this fish, I got extra opportunity, extra interesting stuffs to look into, and some of these were like a mechanism and the pattern of the yolk sac absorption. So the first time I could see in this work, I could see that how the angiogenesis in the yolk sac is happening, how rapidly angiogenesis of the blood vessel formation in the yolk syncytial layer is happening, and how this, the blood vessels are helping in digesting and bringing out the nutrient from the yolk sac to the liver. That fine dynamics of the yolk sac absorption I could study, and then we report this. And then eventually we learned how the development of the stomach, as well as how the pyloric sphincters or cardiac sphincters looks like. So here I'm showing here, my cursor is moving, right? So in the second panel, second picture, hello, am I audible? Hello.
Yes.
Yes, you're.
So here my cursor is moving, right? Like you can detect my cursor here, right?
You can see the cursor moving.
Yeah. So here I could see the cardiac sphincter. So clearly, different zonation. So this is the esophagus and this is the stomach. So clearly these goblet cell patterns are, indicating the cardiac sphincter. And then this is here in the E section, E micrograph, we can see the pyloric sphincter. And then in that paper, we could learn about the function of the pyloric sphincter. So we came to know that pyloric sphincters separate the liquid chyme from the solid, semi-solid in the stomach. The chyme is semi-solid in the stomach, and then the pyloric sphincter allows only the liquid chyme to go into the intestine. That kind of biomarkers we could see, histology-based biomarker we could see in this paper. And then eventually, out of 8 in the previous one, in this work, we could enlist around 20 biomarkers, histology developmental biomarkers. And then eventually we are thinking of, bringing some interesting quantitative biomarkers as well in our coming work on the ontogeny. So we are working on ontogeny of the what is called the rainbow trout in response to temperature. We'll be bringing this story very shortly. So while working on the golden mass here, I came across some of the interesting postprandial biomarkers as well. This postprandial dynamics we could see in the gastrointestinal tract, and then those were the luminal distension as well as the fold, intestinal fold, dynamics. And then some were supranuclear vesicle dynamics, the supranuclear vesicles, which we can see here in the posterior intestine. And my cursor is moving here. You can see these are the supranuclear vesicles, and it doesn't remain the same in numbers as well as in size during the daytime. In 24 hours, it changes with the, the postprandial shift in the shift in postprandial timing. So we could that time we could think of identifying some of these markers as histology based appetite markers, and we're planning to report this as another novel histology based appetite markers in fish. So eventually I got an opportunity to work with another group of people. So this was like comparison of the developmental ontogeny of the golden mass cell larvae from different families, like wild captured brooders, as well as wild captured progeny of the wild captured brooders and the captive rear brooders. So here in this work, what we could see was, so there was no difference in the timing of development, but there was a difference in nutrient assimilation and deposition. So in this study, what we found was wild captured brooders progeny were very active in protein and lipid absorption. So in the top panel where I'm moving my cursor is, this is the indication of the protein absorption in terms of supranuclear vesicles. This is from the captive one. And then down below, I'm showing again in the G, what is called the photomicrograph G in this, what is called the right panel of the image. So here these, the supranuclear vesicles are, more in numbers, in density, as well as the size. Similarly, the lipid absorption was also more in the wild captured, the larvae from the wild parents compared to the captive parents. And then we could see the dynamics of nutrient in the liver. The left-hand side image is from the liver. So the dynamics, this is the nutrient deposition dynamics, the evacuation of the hepatocytes. So it was happening so quickly in the wild larvae compared to what is called the captive larvae. So in another work, I got an opportunity to work on, how some of the gastrointestinal physiology changes in response to methanotropic bacteria, or the methanotropic bacteria incorporation and the rainbow trout feeding for replacing the fish meal. So we could see here the protein absorption pattern was changing with the incorporation of the bacterial meal. So more and more bacterial meal when we incorporate, when we increase the incorporation of the bacterial meal, the protein digestion ability, luminal protein digestion was decreasing, and what is called the intracellular digestion was increasing, and corresponding to the decrease in the protein digestive enzyme activity in the intestine. And then similarly, the lipid in the hepatocyte was increasing in response to the bacterial meal. So these were the biomarkers we could develop over the period of time, 6-7 years, how we can use in other subsequent studies, coming in the future. So some other people who are working on the digestive tract ontogeny are the group of people from NBFGR Lucknow, and then they published 2 papers on digestive tract ontogeny of the catfish Omphagbimaculatus and Heterobnistis fossilis, and we are following them. And then, until now, very limited papers, limited work, limited baseline data generation on the, candidate aquaculture species ontogeny work has been published from India. So I came to know, during my work, I came to know one of the actively working professor from IRTA Spain, and then I find this guy very inspirational. His work I followed, and the kind of the detailed study these people are reporting on the what is called the larval nutritional physiology, as well as the ontogeny work. And then this was really inspiring to me. And then I want to share this what is called the Google Scholar page of the Professor Henry Gisbert, so that the student who are interested to work on the digestive tract ontogeny can visit this profile on Google Scholar page as Henry Gisbert. They can search and find out the work he has done, the enormous amount of work he has done, during his time, and still, people can go there and work. So moving on to the reproductive physiology work also, I got an opportunity to work during my PhD program in Texas Tech University. After joining, so in that, my PhD dissertation work, we looked into the gonadal sex differentiation, or the gonadal sex ratio, and the pubertal development in zebrafish in response to thyroid endocrine system. We inhibited the thyroid and we supplemented the thyroid, and we looked into the gonadal sex differentiation, and we find out that the inhibition of the thyroid lead to the female formation in zebrafish, and supplementation of thyroid, skewed the population towards the male, and then skewed what is called, induces the precocious puberty in male, but not in female. So that kind of work I started in reproductive physiology in my PhD program, and I got to see some of the markers, some of the identification, and making myself familiar to the gonadal histology and identifying those organs, systems, how it works. And after coming back from there, I got an opportunity to work with people in DCFR, Dr. Akhtar, CG, and then we worked in photothermal manipulation and its response on gonadal maturation of golden mouse hair. So this was one of the work on histology-based reproductive physiology study in DCFR. So another type of work, what we are doing on the development, what is called reproductive biology, is the study of the seasonality of the gonad. So we did the gonadal seasonality study on the golden mouse hair. And then we find some of the interesting stuffs, like how gonadal seasonal changes is bringing the germ cells shift in the physiology, what is called the histology of the germ cells dynamics. And then based on the what is called the germ cell dynamics shifting, then we can tell the fishes are going to spawn, they are in the regression phase, they are in the developing phase. And then this was one of the work we could do in DCFR. And on another species, we worked the annual seasonal changes in the reproductive gonadal status of the snow trout. And then lately, very, very recently, we got an opportunity to see how beta glucan is affecting the reproductive performance of the fish. So our model species in this work was Torputitora, golden mouse hair, and then in this work, this paper is in review now with this opportunity to work. In this paper, what we could see was, the progressive changes in the atresia of the ovarian follicles. So this was the first time I got an opportunity to see the progressive atresia, and then I was when the atresia progressed to the end, the follicular remains look like a post-ovulatory follicles, and then it was so confusing whether this is a post-ovulatory follicle complex or post-atretic follicle complex. We had to go back and detect this as a post-atretic follicular complex. So this work helped us to identify the gonadal anomaly study through histological lens. So moving on to another thing called histopathology. So histopathology, how can we use the histology tool to study the histopathology? And then I find this subject very interesting because during my PhD program, my professor recommended some of the papers and some of the books that I can refer, and I can understand the histopathological changes, how the normals, the histology or the cellular structures looks different from the the structure, what is called the tissue or the cells, which are not normal. So I got to see this book, Histology and Histopathology. If some students or some faculty, if they are interested to go on to study the histology and histopathology and incorporate this tool in their research, and then they can follow some of the books which I have mentioned here. And these are the books I got inspired to work on these tools of histology. So this is the down below I'm showing histopathological assessment of the gonadal tissue in the wild fish. This is written by the V.S. Blazer. Her name is Vicky Blazer. And then she is a professor I have seen working in the two universities in the US. I think she is already retired now. I have seen archive of literature published from her side. She had multiple collaborations, and then her was mostly on the histopathology of the fishes. And then gradually I went, and I find this interesting, histopathology, I find this interesting. I went through the literature she developed and she published, and they are very interesting to see. If some students, they are interested to work on the histopathology as a, the main subject in their career, then they can visit her profile in Google Scholar or the ResearchGate, the Vicky Blazer, and then they can access to her work on histopathology. And then another interesting paper what I saw was a reference paper. A reference paper was brought by some experienced professors from the US. Mostly are from US, and some people are from the Germany and Switzerland. This paper's impact factor is 1.9, not much, but the contribution and collaboration is so impressive. People from around, this many collaboration they did, and they brought a very nice guideline paper, a reference paper to work on, to understand, to study the histopathology in fishes. This is the one of the Bible paper, to study the histopathology in fishes. And that's why I'm showing here the students who are interested to learn this subject. They can go through it, and they can find this interesting. So this is one of the interesting thing I could see. And I found and I went through it and I learned so much from this paper. So in India, until now, we do not have any guideline or reference paper of this kind, and in future we should be working together and bring out, some of the impressive work like this. So another interesting literature, what I found was, the resources material for the training programs, the materials, and the institute in USA where they offer the histopathology and the pathology work in fishes. So the students who are interested to to choose this as a career in their life, then they can go through this paper and they can find some interesting. This was brought by that many number of the experienced collaborators from United States. And then these are the subjects they are discussing here, the courses that students can go through. The histology and histopathology courses are offered by some of the institutes in US. The universities or the institute, they can go and learn about the histology and histopathology. These are highlighted in the yellow, what is called the yellow highlights. So these are the very, what is called the useful information that I want to share this with the the students who are interested to study histology or histopathology. So in the line of histopathology, we get an opportunity to work with collaborators from the pathology division in our institute. And then in this study, what we saw was the histopathological response to Aeromonas salmonicida injection in common carp. And then this was the first, the first histopathology study I did in DCFR. And then interestingly, some interesting finding we got, the sinus, what is called the congestion of sinusoids, the red blood cells, with the cursor I'm showing here with the what is called the solid arrow is indicating the sinusoid congestion with the red blood cells. So when you inject the fish with Aeromonas salmonicida, you can see the kind of degeneration which is linked to the which is linked to the red blood cells. So red blood cells get into the tissue, and then it deteriorates everything, like starting from the red blood cell everywhere in the gut. This is the the infection caused by the Aeromonas salmonicida, and this is the histopathological response where the red blood cell is everywhere in the intestinal fold, and then the mucosa of the intestinal fold is completely gone, completely degraded. And this is the cursor I'm moving here to show how it affects the gill. So this is a lamellar fusion, the indication of lamellar fusion, as well as what is called the hyperplasia, or the hyperplasia of the secondary epithelium or secondary lamellae. This kind of histopathological markers we are trying to develop to working with different kind of pathogens. So this is another study we can do in DCFR. So this is a response of Edwardsia latarda. This is a bacteria in grass carp. So after injection, we could see the infection, the pathology of the gill. Similarly, here also the lamellar fusion was seen, and then this is epithelial hyperplasia, and then clogging of gill was clearly visible in the histopathology, and then complete degeneration of the submucosa here. I'm moving the cursor here. The complete degeneration, selective degeneration of the submucosa or the connective tissue. The mucosa is completely intact, the muscular layers and serosa are completely intact, but the submucosa is completely gone. So this is here I'm moving the cursor on the kidney tissue. So here we can see the tubular degeneration, especially what is happening, specifically what is happening here is the basement membrane and the submucosa is completely degrading in the first, in the initial stage of the infection. So gradually it leads to the degeneration of the tubular, what is called the renal tubules. So this kind of work we are doing on histology, histopathology, and trying to develop biomarkers so that we can one day we will be able to, we'll be able to write some guideline papers or what is called the reference paper of Indian feces. We are trying to generate the informations from the Indian tropical feces, because the information on our feces are very limited until now. We have seen that we have to work on this baseline data generation, how they exactly look at the the tissue level, the phenotypic response, in terms of the histology or histopathology. We have to really see and then report and then archive this information and publish the guideline papers in Indian feces. So this can be a very good reference work in the future. We are working towards it. I don't know how long it will take to bring out such publication like other people have done in the the first world countries. So this is another work here. I'm showing that how the case study of how rainbow trout is affected, in some of the farms, how this pathological condition was seen. Here in this work, what we could see was the selectively or specifically only the pancreatic tissue was degenerating. The necrosis of the pancreas, necrotic degeneration of the pancreas was seen in this study. So this was one of the first time histopathology work we did in our institute. So I'm very thankful to my mentor, my professor who taught me histology, ABCD of the histology and histopathology during my PhD program in Texas Tech University, Dr. Rinaldo Patino, Dr. James Carr. I worked with him to teach the animal histology course and the comparative physiology. So I got an opportunity to learn, the what is called the histology at a broader level, like animal, how fish histology or how fish microanatomy is completely different from the animal microanatomy. So the professor, Dr. James Carr, he taught me, and then experimental histology, histopathological response was taught by my professor, Dr. Rinaldo Patino. I'm very much thankful to some of my professors from India, Dr. G. Venkat Swarlu. He's a joint director at ICAR-NAM, Dr. A.K. Pal, who supported me during my master's program and always encouraged and inspired me to go further for my PhD program. Dr. P.K. Pradhan, who is a principal scientist in ICAR NBFGR, who inspired us during our undergrad times, and then he inspired us to go for higher studies. And he was the first one to develop the interest of science in those days. So we were inclined to learn about the science, learn about the research, and how science and research can be an interesting career in our life was taught by the professor Dr. P.K. Pradhan. So I'm very much thankful to our present director, Dr. Pramod Kumar Pandey, and then the previous director, Dr. Devajit Sharma. Dr. Devajit Sharma, he provided me a platform of histology in DCFR so that I could initiate my work and I'm liking it. To work on the histology since then, 2015, and I got an opportunity to work with many collaborators from the institute and outside of the institute, like within the ICR. And then in future, I'm hoping that more people will come, and then we can work in collaboration. And my previous director, Dr. A.K. Singh, he also inspired me to take this tool and take this science as a career in my my profession. So I'm very much thankful to my active collaborators like MS Akhtar, Dimple Thakuria. They got an opportunity. They gave me an opportunity to work with them. Dr. C.G. Alexander, Biju, Sam Kamalam, Rajesh M. I work with these people. Dr. Rakhi Kumari, Ritesh Tandel, he's from pathology. Dr. Adil Bhatt, he's from pathology. Siva, with Dr. Siva, I got an opportunity to work on, the gonadal ontogeny of golden mass here, and how sex differentiation, what are the, how histological events are happening in the gonad, how germ cells are, first how they are dividing through mitosis, and how they are, undergoing the meiosis, and how the sex differentiation is happening. I could study along with Dr. Sivasi, and I'm very thankful to Mr. Pathadasi, the technical staff, who always helped me during my initial work in DCFR. And I'm very much thankful to my student Manish Kumar Dubey. He helped me like my right hand in doing my histology work, and then he is a totally dependable guy, very serious, very much responsible. And then we both are developing what is called curiosity, interest in histology and histopathology, and then we are discussing together. We are learning and bringing out the more interesting stories in the future. Other students who worked with me are Mrs. Anu Sharma, Miss Rini Joshi, who is currently working as a PhD student, and then Miss Bandana Pandey. She came to our lab as a master's student, and then she really worked very nicely, and very good dedication she showed, and she did a histopathology work in our lab. So am I audible to you guys until now?
Yes, you're fantastic.
I want to conclude here, and then if you guys have any questions, I'd like to answer happily.
Yeah, thanks very much, Dr. Prakash.
One thing I'd like to tell here is why I was interested to give this. The talk was, the histology or histopathology is a very conventional tool. People are not using it because nowadays people are, more interested to go for, molecular tools to study science, or, some other advanced tools people are using. But I find this tool and technique very interesting, because we can see what is happening inside the fish, what is happening inside the fish at the tissue level or the cellular level. And then while doing my work, how I enjoyed the work was, when we slice the fish. So we do serial section, and then we slice the fish. And then every slice of fish is like, pages of the book. And then we can read how things are happening inside. We can, observe those under the microscope, and we can we can observe this as reading of, individual pages of the book. So this is very much interesting subject, and I want, young generation that, I want to inspire the young generation. I want to tell the young generation that this subject is very much interesting, and you should go for your higher studies in some places where you can study, histology as a, career opportunity for you guys in the future. So my main objective to, talk about the histology was, to develop interest in people who find this, a little bit interesting. So I don't know how much successfully I could deliver or how much. I could,
You did very well, Prakash. So I never seen any talk going in that depth into the fish, histology. So it definitely looks very informative, and also probably the first time we can have a talk here, actually showing the important papers and where to look for, the best information. So I think this is probably one of the probably the best talk for students, and definitely that the presentation is made to the purpose to generate interest in people who are looking forward to it. So thanks everyone for joining in, and thanks for Dr. Prakash. So before we go to discussion, just give us a feedback about today's talk. So I have provided the link in the chat box. And those who joined later in the talk, I just want, if you have any questions, please post your questions in the chat box. This is just for all of us to understand what you're asking, but you will be given a chance to.
Sir, namaskar. मैं पाल सर को देखा अभी. Sir, namaskar.
Sorry, can I finish what I will finish what I'm talking, and then yeah. We are also looking for expanding our coordinator network. So if any of you are interested to join us in our initiative, you are free to come and just get in touch with one of us. So I'll go to these questions. I'll give a minute for everyone to think about it. While you're thinking, I will ask a couple of questions from my side. If you're okay, Prakash.
Yeah, no problem. Yeah.
Yeah.
I'll be happy to answer your questions.
Yeah, so just for the benefit of the people who are outside the world of histology and those who are new in the field, for the students and everything, I just want to ask this question: what will be the high-level application of all these studies? How these outcomes can be used to bring a real change in what we are doing at the moment. And maybe if you can give me some examples, like in probably more in the layman terms, how these studies, how we can bring a change in our current practice, current understanding of how, whether it be in the aquaculture or, hatchery or production or whatever. Whatever you can think of, probably it probably could be in the conservation.
Yeah, I got your question. Yeah, I got your question. And then, yeah, that's why I brought this subject, because I found that, the data generation, baseline information, the data repository, using this tool is not being done so much in our country. That, when I was looking for the literature available on the gonadal seasonality of Indian fishes, like marine to the freshwater fishes, these informations are very much limited. So, this is a very much a visual tool to see, the spawning seasonality of the fishes, and then, the germ shell development pattern. So if we want to capture a fish from the wild, and then we want to develop a breeding techniques or breeding technology, if we want to bring out, and this tool can be used to see the gonadal changes, and then how gonadal changes, as well as how nutritional changes are happening inside, during the corresponding shift in the gonadology of the fishes. Then this tool can be used, and then, it can be a very good information there. So it can be a very good tool to study or, develop a breeding techniques for any kind of wild fish. So another thing,
improving the strains, or
yeah. So yeah, lately I was looking for the gonadal seasonality study. So done through the histology, and these literatures are very limited from our country, very much limited from our country. Most of the gonadal seasonality works are done mostly based on the GSI or HSI, or what is called it, the condition factors and those things, the eggs availability and those things. And we can incorporate in histology as a tool to study, restudy this gonadal seasonality of the fishes, and then we can find potentially some interesting fish that we can bring from the wild and try to breed down captivity. And that there we can use this technique. Another place where we can where the technique has application is to study the larval nutritional physiology. So when the larvae is how the larval development takes place in terms of the nutritional physiology, and when they are ready to feed on the exogenous feeding, when they are ready to wean to the the pelleted feed or the what is called artificial feed from the live feed, that kind of study we can do with the histology tools. And another thing, we can go further advanced form of histology or histopathology by incorporating immunohistology or in situ hybridization or electron microscopy to further bring the more specific changes in the histological response in this kind of study. So did I somehow could able to answer your question?
Sorry, you become inaudible. But can you speak? Just your voice is very low. You can still hear me? Hello?
Yeah, I'm hearing you.
Yeah, it came back. Okay, no problem.
Internet, yeah, internet also.
It's breaking.
Sometimes it is going off and sometimes it's coming. The wave is not consistent.
Okay. You have just two more questions before I go to the audience. One of my question is, I saw a few of your papers are on endangered species, right? So I'm assuming probably the reviewers may ask, because this will include sacrificing fish. So so I guess most of your work probably you have fish in captivity for the research, I guess, or are you really sacrificing wild populations for the study? You probably get some questions from your reviewers.
Yeah. The sample size, actually, we are using a limited sample size to study those species.
Yeah.
So what alternative I think we can use is using the surrogate species to study those basic information, basic science for these species. We can use like a common carp as a surrogate species in place of golden mahseer. Yeah. And then we can see the what is called phenotypic response study we can do there by we can use that way.
Yeah. So just by looking on
so why I told understanding the reproductive seasonality and then develop the breeding techniques. One of the colleague from our institute, Dr. M.S. Akhtar, he did the reproductive physiology. He studied the reproductive physiology of the fishes, this golden mahseer, and he brought into the captivity and he successfully bred the fish in the captivity. So I got an opportunity to work with him, and then finally he developed the technique. So in the similar way we can work on other species also, and then we can produce the seed artificially that the fishes, this kind of endangered fishes, will no longer be endangered in the future.
My last question is, there were a few papers on larvae, ontogeny and kind of thing. So I'm not an expert in the histology or this thing, but I presume, when you study larvae or, even stages at very young stages, the development is so fast, so you probably have to take samples like every 1 hour.
Every day we have to take,
yeah.
Or every 12 hours is good, but every day is okay.
Yeah.
To take the samples to see how quickly.
I got you. Yeah. So I'll pass my baton to the audience. There's one question, first question from Femi how can we explain situation?
Deepak, before going into the question, I'd like to highlight why this ontogeny is totally different from one family of the fish to the another. There are two different kind of developmental pattern. So one is called the precocial developmental pattern of the digestive tract, and another is altricial pattern of the development of digestive tract. In a precocial pattern, the digestive tract formation takes place prior to the onset of the exogenous feeding. So when the yolk sac is still there, most of the development takes place. So in this species, when the larvae exhaust their yolk sac, they are very much developed from the internally. They are very much developed so that they can accept the artificial feed, and their larval survivability is more than 90%. So some of these species which the example of this kind of species are golden mass. Most of the cold water fishes, they follow this kind of pattern of development. And then the marine species, mostly tropical species like IMC or the marine species or the catfish, tropical catfishes, they follow the altricial pattern of development, where the digestive tract developments still will be in a will be going on after the onset of the exogenous feeding. So their efficiency of digestion and assimilation after the onset of exogenous feeding is not that much good. So that's why the larval mortality is very much high in this kind of species.
Yeah.
So in future, if we can manipulate, if we can manipulate the larval developmental ontogeny, and then try to make the altricial species, try to try to transform the altricial species in the precocial species, and then we can really enhance their survivability. So this is why this kind of informations are very much essential to understand their developmental pattern.
Yeah, that makes sense. So there's a couple of questions in the chat box. The first question is from Femi. Would you like to come and speak directly to Prakash, Femi? Are you there still? Yeah, you're there.
Oh, thank you so much, Dr. Prakash. Thanks so much for this.
You can speak, Femi.
Can you hear me? Am I audible?
Not audible to me.
Oh, can you hear me now?
And just need to be a little bit louder.
Not audible to me.
Oh, it's not audible to you?
I think maybe I will read out the question because not audible. How can we explain situation where we have severe macrovascular steatosis and moderate vascular congestion and very mild periportal inflammation in liver histology of tilapia after fed some nutraceutical? Can you read, Prakash, in the chat box? Can you read?
Yeah. I'm following your question. Like steatosis, it's kind of a lipid deposition, right? So it's a hepatic vacuolation or the lipid deposition in the liver. So sometimes it can be an indicator of the nutritional status of the fish when the fish is in normal condition, and sometimes it can be a pathological changes. So depending on the situation or the kind of the treatment we give. So in one of my few instances I have, like when we study the seasonal shift in the nutritional status of fish. So in golden master, we find interestingly the liver nutritional deposition in terms of the fatty deposition or the lipid deposition, and liver was very much seasonally different in the case of male, but in the case of female it was not that seasonally different. So this is a normal lipid deposition in the liver. So in another instant, I'll say, like, when I zebrafish. So am I audible now?
Yes.
Okay. When I
No, not now.
The thyroid status by inhibiting the thyroid by methimazole treatment. So in that case, there was a lipid deposition in the liver. So that is a pathological condition. So what is called in that case, what I observed was, their feeding behavior was very much decreased. Still, the lipid deposition was there, meaning their metabolism was not normal. The metabolism was completely retarded. So that's a pathological condition. So,
Yeah.
so lipid deposition in liver can be, can be an indicator of pathological condition as well as a normal nutritional deposition, but it depends on the situation where you are using it.
Thanks, Prakash. I think I may have answered Femi's question. One question from Gokul is, can you explain what are the difficulties in gonadal histology and tissue histology at procedure level?
That's an interesting question because definitely we are also.
Sorry, you're not audible, Prakash.
This is an interesting question.
Yeah.
So now am I audible? So when we are doing the male gonadal histology, it is very easy to cut and, there is no problem. When we are doing the female gonadal histology, and if they are like, it has more yolk content. When the yolk content is more, and then, the sectioning is very much difficult. So we are trying to develop a fixative that is good for fixing the ovarian samples. So we are working on it. We are trying to formulate some sort of fixative that can give efficient sectioning of the female gonad. So until now we have been using the IMC gonad also, the gonad which has a smaller eggs. So in that case we can use formalin NBF 10%. So when the yolk content is more, we are using the Bouin's fixative. So but the Bouin's fixative is also not the absolute fixative that can be used for the ovarian fixation, but we are working on it. Eventually, maybe in 2-3 years, we'll try to bring out some sort of formulation. And so that's the thing. I think his intention of question was that one, if I'm not wrong.
Yeah, Google, if you have any further questions, you can come and speak. You can mute.
Sir, I am audible?
Yes.
Yeah, you're audible.
Sir, what are the difficulties you have, sir? You do histology studies, sir. That time, what are the difficulties you have?
So what are the difficulties that we face doing histology, right? While doing histology.
Yes,
So in histology, there are like some equipment we should have. One is the processor. Tissue processor has to be there because the constant temperature of the paraffin should be there. When the paraffin temperature goes up, when we do not have a temperature control system of the paraffinization process, right? Then the temperature will go up, and paraffin will the temperature of the paraffin, if it goes high, it will fry the tissue. So if the tissue is fried, then there will be a anomaly. There will be a similar corresponding changes in the tissue level. So that kind of the problem is there. But sectioning is not a big deal. Sectioning we can do easily. And then if we have a problem in sectioning, and then we can put the tissue in -20. And if we put the tissue in -20 and take out the tissue from -20 and cut, the sections comes really nice.
Okay.
And then staining also, then you have to choose the staining, what is called precisely, because the hematoxylins available in the market are different. You have to identify which hematoxylin is good for staining, which hematoxylin gives, the good staining, so that some hematoxylin, it gives a good staining, but it will not allow the eosin stain to come. So that, in that case, eosin and hematoxylin, when we are doing the basic staining, eosin, we are doing the hematoxylin stain first, and then we will wash, and then we do the eosin staining. But sometimes if the hematoxylin is not good, the eosin stain doesn't get into the tissue. So we cannot identify the tissue separately. So that kind of, the staining identification, what is called the stain specification, we should know priorly.
Okay, sir. Thank you.
And some of the specific stains are also there, like PAS stains for carbohydrate staining or goblet cell staining. So immunohistochemical study staining we can do with antibodies, or in situ hybridization, that will be much cheaper than the immunohistochemical, because antibodies are quite expensive than the the RNA based probe designing and all.
Yeah, so I think the next question is from Lavdeep Sharma, and I think this is probably a typical question for probably from most of the students who have no experience with histology. So he's asking, can you please give an idea about the methodology to be followed for studying gut and gonads of cyprinids? So I think maybe there'll be more people who actually came to know how we do histology, like basic things.
I can send them, no problem. Methodology for studying the gut and gonad is a very basic methodology which is available everywhere. So you can use NBF 10% for fixing the tissue, and then you can process in series of 70%. You don't have to, alcohol people are reporting 40%, 50%, that kind of alcohol people are reporting. What we are doing is 70% ethanol 2 times, 95% ethanol 3 times, 100% ethanol 3 times, and then xylene 3 times, that kind of we are using for dehydrating the tissue. And then after putting in xylene, we go for a paraffin infiltration, and then put paraffin in three sets of paraffin, like 45 minutes each at 60 degrees centigrade. So paraffin 1, paraffin 2, and 3 times we do, and then we go for blocking. So we make blocks in tissue embedder, and then after blocking we section the tissue, and then we stain. The first step of the staining is deparaffinization. In the processing we do block in the paraffin, and then while staining, the first step is deparaffinization. So first we have to put the section in the xylene, 3 rounds of xylene, 100% ethanol, 95% ethanol, then alcohol 70%, and then in distilled water. And then we can go for staining with the hematoxylin, eosin, after washing in alcohol and water. And then finally, we have to, what is called, re-dehydrate in series of alcohol and xylene, and finally cover slip for making a permanent slide.
Yeah, that's a pretty quick overview on how you do histology.
This is a very general, and then you'll find literature everywhere. So specifically, if you want to know the literature we follow, the protocol we follow, we can share with you, no problem.
Yeah, can you type your email address in the chat box, so if anyone want to contact you directly, Prakash?
Yeah, definitely, I'll.
Yeah, so there's
yeah, I can be reaching that email.
Yeah, so the email address is in the chat box if anyone of you want to contact Prakash directly.
Basically, if you want to ask any questions, protocol questions or any kind of thing, I can answer your questions.
Yeah, And there's Reshma asking whether the recording is available. Yes, recording of this talk will be available in our YouTube channel after 2 weeks. So that's the standard thing we do. Just follow our YouTube channel and you'll be notified when there's a new talk available to watch. So I have put one more link, once again, the feedback link. Please provide your feedback and I will send them to Prakash. So if you have any good things, nice things to tell, I will send only the good review to Prakash. But I think there will be really good ones. Yeah, if any other questions, please unmute and you can ask if any of you have a question or any comments, anything.
Hello.
Hello, Dr. Prakash. Hello, Dr. Prakash. Hello.
He's still here, Prakash? Yes.
Yes. Hello.
Hello, Dr. Prakash. I'm happy to see you and your nice
Sir,
talk.
namaskar.
Namaskar. But I have one
सर
query.
आपके आशीर्वाद से सर जो कुछ भी कर रहा हूँ सर आपके आशीर्वाद से.
I have one query. Is there any technique developed so that we can quickly do the histological diagnosis, like in case of the human being?
Until now I have not seen any, sir.
In fisheries, there was such development, no?
I don't think so, sir. I have not come across those things, sir.
Okay. That's so.
Yeah, one of my colleague was asking one day that we should develop that kind of technique, but I don't know.
Because
So
in the field, I am facing that many of the diagnosis takes a lot of time. At that time, fish dies. So that's why, is there any quick method so that we can diagnose the disease? So, but if we send the sample, it will take at least 1 to 2 days.
definitely in future we'll try.
Okay, you should develop that type of technique, a histological technique for diagnosis in any type of disease, yes. Or any recent technique in other countries also, if it is there, that also you can try for. Oh, thank you very much. I am happy to see you after a long gap.
So much, sir.
Yeah.
Thank you so much.
Okay.
Thank you very much. It's, after a long time, I can see you.
Yeah.
Anyone else have a question?
Can you hear me very well? This is Dr. Follic from Nigeria. Am I audible now?
So I think that's everything today, I think. But if you have questions, feel free to contact Prakash. He is always available to give you any assistance or support. Yeah, so join us after 2 weeks. So there'll be, we have an ongoing webinar series. So there'll be always some exciting talks coming up. So keep following us. We have a Facebook account where you can follow our page. We have Instagram and we have a WhatsApp group, Telegram group. So you always send notifications when there's a new talk is coming. So I once again thank you everyone for joining in, and thank you, Dr. Prakash. Great to see you, and we'll bring you back again for another talk.